Thanks very much for asking me to introduce the two fantastic students who are going to speak at today's event. I'm so happy to do that, as you said, as I am indeed myself an alumni of Sheffield University, graduating in 2001 from the biomedical science course. And then fate drew me back to Sheffield, where I've been working in the neuroscience department since 2006. And in 2010, we had the opening of the Sheffield Institute for Translational Neuroscience, or CITRAN, which really was a huge step forward in the neuroscience initiative taking place at the University of Sheffield. And I've got some photos everybody should be able to see on the screen now, showing the CITRAN building with the large letters CITRAN made of Sheffield steel. And you can see it pictured at dawn, at midday, and also at dusk. So always a blue sky or a purplish sky in Sheffield, as I'm sure you all remember from your time here. And that's really to signify the groundbreaking and in many cases world-leading research which is ongoing at CITRAN. Doesn't ever stop. And so we're working around the clock on various neurological conditions. And the research at CITRAN, and the reason why CITRAN is so special and such a unique institute to work in, is because it houses within it so-called basic scientists like myself. So I'm a biochemist scientist by training, and I work very closely and alongside clinician scientists like Tom and Simon, who you're going to hear from today, also from clinicians who are focused solely in the clinic, and also then with other research staff, research nurses, also postdoctoral scientists, PhD students, research assistants, and also bioinformaticians and a larger group of people who really enable the collaborative work that goes on at CITRAN. And that means that we have really quite a special environment that enables us to really do bench to bedside research. And a lot of places talk about bench to bedside research, but actually in CITRAN we are in fact really doing bench to bedside research. And in fact, one of the programs that I worked on as a postdoctoral scientist in the laboratory identified a drug which might be helpful for Parkinson's, and now it's being used in clinical trials, and Tom will talk a little bit about some of the work associated with that later. So it really is truly an amazing place to work, and it's a unique environment, not only within the UK but around the world, that really enables this truly translational research to happen. And CITRAN had its 10th anniversary at the end of 2020. Unfortunately, we couldn't do the large event that we had hoped to do because of the pandemic, but we still featured some online events. And the final picture that you can see in the bottom right-hand corner is actually an event which happened just squeaked in before the pandemic really hit in February of last year, when CITRAN was awarded the Queen's Anniversary Prize for its work in neuroscience, and particularly in aiding neurodegenerative disease research. And I was very honoured to be one of the few people from CITRAN that was able to attend that event at Buckingham Palace. And for the eagle-eyed people amongst you, in the bottom right-hand corner of the picture, then that maybe you can see with my pointer now, the turquoise stripe of a dress, that's actually me sitting there at Buckingham Palace at the Queen's Anniversary Prize when we received that award. So it really is a very special place to work, and a great pleasure for me this evening to be able to introduce to you two PhD students working as clinician scientists at CITRAN, who I've had a hand in various guises of supervising throughout their PhD. So Simon's work focusing on Alzheimer's disease and the batteries of our cells, the mitochondria. That is my area of research, and so really in scientific terms, I would be a fully paid up member of the Mitochondriac Society. I think they're absolutely fascinating parts of our cells, and my research team really focuses on understanding how they go wrong in various neurodegenerative conditions, and then indeed, can we target them? Can we boost their function as a treatment for various neurodegenerative diseases? And so I'm just going to leave you with a final thought before I pass on to Simon directly, and that's so over the last 10 years of Citran being open, we've really made some fantastic scientific discoveries in various different neurodegenerative disease conditions, and it was fantastic in the year of our 10th anniversary to be awarded the Queen's Anniversary Prize. But we don't stop. We're still striving for excellence and for further groundbreaking research and breakthroughs in various neurodegenerative conditions. And that's why we are also now in full-on planning stages for a Citran 2 building to be built alongside our current Citran building. And that will enable us to keep at the forefront of the groundbreaking research around the world, enable us to grow our team of scientists and clinician scientists and researchers working on making breakthroughs in these various neurological conditions. So with that, I'll pass over to Simon, and I'll let him tell you about some of the work that he's been doing with his PhD. Over to you, Simon.
Okay, thanks, Heather. Right, so I'll just start my sharing my slides. Put this to this side here. Okay, so my name's Simon Bell. I'm a neurology registrar, and I'm like a lot of people now in this room, as well as I am also a Sheffield University alumni. Now, as I've actually recently completed my PhD this year. And my talk's mainly going to focus around how the brain uses sugar and how this might help us to understand or develop new treatments for people with Alzheimer's disease. Now, I thought I'd tell you a little bit about myself before I go on to discuss the topic. So if you can't tell by my accent, I am a Yorkshireman. I'm originally from Leeds. I have quite a varied sort of training. I trained in Bristol originally for medical school, then Gloucester, then Brisbane in Australia, which this picture at the top here represents. And then I moved back to Yorkshire and I did junior doctorate in Leeds. And then I moved to Sheffield originally in 2013 for many of the reasons Heather's just spoke about. I'd heard about the fantastic sort of neurosciences research that goes on here, and I really wanted to be part of that. My research mainly focuses around metabolism, or how the brain uses different metabolic substrates in Alzheimer's disease and how that changes through the condition. And I was very lucky enough to have a PhD funded by Wellcome to study this, which, like I said, I've just finished this last couple of months. And I'm now working as a clinical lecturer, which is a job in which I'm a neurologist or a medical doctor for 50% of my time, and the other 50% of my time I work in the university. Now, two important facts about me: I am a huge cricket fan, as I'm sure you're all very well aware. This field here is Yorkshire Cricket Ground, a very happy place, especially if you're a cricket fan in 2019. And I also play the trombone in a local Sheffield orchestra, and anybody who plays in orchestras will understand the sort of stereotypes that trombonists are sometimes tarred with, which I think this little diagram at the bottom here represents quite well. So, Alzheimer's disease is the most common cause of neurodegeneration with it throughout the world. It affects roughly 800,000 people in the United Kingdom, and up towards sort of 30 to 50 million people worldwide. It's an incredibly expensive disease, and by 2030 we reckon $2 trillion a year will be spent on looking after people with Alzheimer's disease. But the costs are not just financial, as I'm sure you're all fully aware for people who've looked after people with Alzheimer's disease, as both the psychological and social costs of the condition are very extreme for the families as a whole. As we age, the risk of Alzheimer's disease increases, And at the moment, we don't really have any good treatments for the condition. And hopefully the aim of my research and a lot of other research done in Sheffield is to try and readdress that balance, and hopefully we might be able to develop treatments for the future. Now, classically, Alzheimer's disease was thought to be a problem of just the neurons of the brain, or the thinking and memory cells. And what generally has been shown to happen is that there's a collection of proteins that occur within the tracks of the neurons, or the axons, and the neuronal bodies themselves. Now, these two traditional abnormalities that we saw in Alzheimer's disease were called amyloid, which is a protein that sort of sticks to the outside of the cell and damages it, and something called tau, which forms part of the trafficking mechanism within the cell. And what happens is that the structure of this protein changes, and that starts to break down the trafficking mechanisms within the cell. And as you can imagine, certain neurons can have axons that can be up to a meter in length. So if you break down the trafficking mechanisms, that's going to lead to problems with the cell. These particular proteins aggregate in particular parts of the brain, and they have a classic characteristic structure which we associate with Alzheimer's disease. Now, as we've learned more about the condition, we've realized that it's not just caused by the buildup of proteins, and potentially how the brain uses sugar is another important factor to the condition. Now, people probably think of sugar as this sort of negative substance that we, as much as we all very much like it, as this picture here shows, but in fact it's the main fuel source for the brain. Now, this diagram here shows the change in sugar metabolism, glucose being another word for sugar, as people develop different memory type conditions. So in a normal brain, we have quite a lot of glucose or sugar metabolism. And when we move to like a situation in which the memory becomes impaired, as you can see, the orange colours have become less in this diagram, and that suggests that the glucose metabolism is lower than in the normal situation. And in Alzheimer's disease, it drops again. And like we see, this gradual decline of how the brain can use glucose as the disease progresses. This is something that happens naturally with age anyway, but it's exacerbated in Alzheimer's disease. So if we could understand that reasoning why, then that might help us to develop treatments for the condition. So one of the first steps we need to try and understand of why the brain reduces its sugar uptake as we age is to understand how sugar actually moves into the brain. Now, sugar gets into the brain through the blood vessels, as it does to most organs, and generally the vast majority of sugar passes through a particular type of brain cell called an astrocyte. Now, this is very different to neurons or the thinking types of cell, and this little green diagram here represents an astrocyte that we've grown in the lab. Now, what astrocytes are, I sort of envisage them as a life support mechanism for the neuron cell, whereas the neuron or cells do the thinking or action, the movements of the body. These astrocyte cells support those in most of their functions, so they're very important to the actual abilities of the neurons to perform. Another word for the astrocyte cell is a glial cell, and interestingly, it's been shown in very clever people that they seem to have a higher number of these astrocyte or glial cells than people who don't have as a high intelligence. And even more interestingly, our brains have much higher numbers of this particular type of cell than what they do of the neurons or the thinking cells. So understanding how glucose is used by this cell might help us to understand why the memory cells don't work as well in Alzheimer's disease, but may also, like I said, try and maybe help us find new therapeutic options for the condition. Now, to study metabolism or sugar usage within different types of brain cells, we need to collect living brain cells to do this. Now, people are generally quite attached to their brain cells while they're alive, and obviously taking samples of them whilst people are alive is incredibly dangerous and can potentially lead to causing disability. And these risks are increased obviously in people with Alzheimer's disease. Now, normally in research, we would turn to using animal models to look at this sort of problem, but the problem with studying astrocytes metabolism in animal models is that the astrocyte cells, or the glial cells of the mouse brain, are very different to what we see in the human. And animals essentially don't get Alzheimer's disease, so we have to change their genome to make them get the disease, which we don't know what effect that will have on the glucose metabolism anyway. So ideally, we need another way to create living cells from people with Alzheimer's disease to study sugar metabolism. And the way that we do that in Sheffield is by taking skin samples from people with Alzheimer's disease, and then we grow a particular type of skin cell from that, called a fibroblast. And this process usually takes about 6 weeks. This is a skin biopsy here, which is growing different cells in a petri dish. What we want to get is to a pure colony of fibroblast cells, which is shown here. And what we can do with that colony is we can then transform that into different types of cells, because every cell within the human body that has a nucleus has the ability to become any other cell. And so what we do, we give the fibroblasts a set of instructions through viral vectors, which changes them into a precursor for different types of brain cells, what we call induced progenitor cells. And this takes between sort of 18 and 24 days. I think you can appreciate the morphology of the cells has changed dramatically here. And then we give them another set of instructions that turns these precursors to different types of brain cells into either neurons or astrocytes or glial cells, as we're studying in this project I'm presenting here. And this is what the cells look like under the microscope. Now, the morphology changes, but we've also done lots of other experiments to look at how these cells function and how they behave in disease. And it's very, very similar to what we see in the cells taken from the human brain before reprogramming. So once we've created our different types of brain cell from skin cells for people with Alzheimer's disease, the next thing we have to do is look at how they use glucose or sugar and how that might be changed with the disease. So one of the experiments I do is something called a glycolysis stress test. And what that involves is looking at different parameters for the usage of sugar by the astrocyte brain cell. Now, I thought that this is a slightly complex idea to explain, so I thought what I'd do is try and use a real-world analogy for this. So this is the diagram that we use to look at the different aspects of glucose metabolism within the astrocyte cell. But I think this is very akin to how humans use money. So this blue box here represents glycolysis. So that's the day-to-day usage of sugar by the cell. So the sugar it needs to perform its normal daily functions. And how I would compare that in monetary sense is the money that we use to go to do our regular day-to-day shopping. This lighter green box here represents something called glycolytic capacity. And that's the maximum amount of glucose or sugar metabolism a cell can perform when it's working at its top function. And again, I think that's akin to the maximum amount of money a person may have available to them to buy a house. So it's the sort of money that you need to put forward for your mortgage. This dark green box here represents what we call glycolytic reserve or sugar metabolism reserve. And this is effectively the difference between the money that you use in your day-to-day life and the money that you would need to buy a house. So I think of that as the money in the bank. So this is if we wanted to increase the function of the cell, this is how much we could increase its glucose metabolism by. Now, as I said at the beginning of the presentation, these astrocyte cells have a role in supporting the functions of the memory type brain cells. And so if your neurons or your brain cells are performing extra functions, then the astrocyte cell has to work harder. So more of the glycolytic reserve or more of the money in the bank is used when the other types of cell within the brain are working at a higher rate. So when we looked at this particular experiment in people with Alzheimer's disease, the astrocyte or glial cells, what we found was that in each parameter, the glucose usage by the astrocytes in Alzheimer's disease was reduced. So in this diagram, the red and the purple bars represent controls of people without the condition, and the green and the blue bars represent people with different types of Alzheimer's disease. And as you can see, the glycolysis rate, or today's sugar usage rate, is lower, as is the maximum amount of sugar that the cells can use as well. But what more importantly we see is this ability to increase your work of glycolysis is very much reduced in Alzheimer's disease. So effectively, the money in the bank for glycolysis is lower in people with Alzheimer's disease, or is lower in their astrocytes at least. Now, this is all very interesting from a perspective of cells in a dish, but what we really want to know is, how does this relate to people with the condition? Now, the skin biopsies I take from people with Alzheimer's disease, we also collect lots of information about their clinical performance. So when people come to the lab in Alzheimer's, to the clinic in Alzheimer's disease, we perform different memory tests on them. And one of those is something called semantic fluency. And this is a memory test that's affected early in the course of Alzheimer's disease. And an example of how we test this is by telling people to tell us all the animals that they can think of within 1 minute. Now, what we see in Alzheimer's disease is that the glycolytic reserve, or the money in the bank, if you have a lower level of this, you seem to score more poorly on this memory test. So that suggests that glycolytic reserve may have some role in a person's ability to perform certain memory tasks. And that's important because it gives us an opportunity to develop new therapeutics by improving the glycolytic reserve for these people. So that's all I was really going to talk about in this presentation. We've discussed how sugar metabolism is affected in Alzheimer's disease, and how different cells of the brain are affected in different ways, and that the sugar metabolism appears to be lower in this particular type of brain cell called the astrocyte, and that if we were able to improve that might improve memory functions of the cell. Thank you all very much for listening. I'd like to thank all my supervisors. Heather was my main supervisor for this project, and my funders. And I'm more than happy to answer any questions at the end of the talks. But I think now what's probably best for us to do is to move straight on to Tom's presentation. So Tom's another doctor and also a clinical scientist that works in Heather's lab, and his PhD focuses mainly on Parkinson's disease and developing new imaging techniques for that. And so I'll finish there and hand over to Tom.
Hello everyone. So I'm just going to set up my screen. And hopefully, yes, good, I can see it. Right, so yes, thank you for your introduction, Simon. So I'm Tom. I'm another clinician scientist working in Sheffield. I'm going to speak to you about some of our research in Parkinson's and what my kind of current PhD project focuses on. To start off with a bit about me, so I'm also a Sheffield alumnus. I graduated back in 2013. Ever since I came up in 2008, I've not left Sheffield. During my undergraduate medical degree, I kind of first became interested in neurology during my clinical placements. And kind of explored it further from there, and then going down a research route further. Since I graduated, I've done all my training in South Yorkshire, either in kind of the Sheffield hospitals or Rotherham. And I started as a neurology registrar and clinical fellow in 2017, and then started my PhD in October 2018, and I'm in my third year now. Outside of medicine, I'm a very keen golfer. No weather will stop me, although admittedly lockdown has. As evidenced by that photo there, I don't mind wind, rain, snow, sleet, whatever. I just enjoy it. And probably one of the only other interesting things about me is we have a cat. She's getting on a bit now. She's turning 22 in just under a week, which is remarkable for a cat. I think in human years she's probably I think approaching about 110 now, or something truly ridiculous. Anyway, so moving on to Parkinson's. So it's the second most common neurodegenerative disorder following Alzheimer's disease. Although we have good symptomatic treatments for the motor symptoms, we have no drug that can slow down the progression of Parkinson's. And there's also a lot of non-motor symptoms of Parkinson's that we don't have particularly effective treatments for. The picture on the right of the screen, so that the figure on the right kind of shows this probably quite old-fashioned perception of Parkinson's, of the individual who is generally elderly, struggling to walk, with the tremor, an abnormal posture, but actually that only shows a very small kind of aspect of Parkinson's. And the figure to the left with their shoulder around them, this is a picture by Dr. Johnny Aitchison, who's an A&E consultant with Parkinson's in Leicester, drew this as a kind of more modern kind of depiction of Parkinson's, looking at a lot of the other factors, things like apathy, sleep, constipation and fatigue, a lot of these factors of Parkinson's which have largely been neglected for many years. Overall, Parkinson's is on the up with our ageing population. The total number of people are predicted to double from 2005 to 2030, and the care costs associated with advanced Parkinson's are really quite high. So there's a lot of imperative at the moment to try and find treatments that can help slow down and reduce the severity of the illness as people progress with it. As I mentioned, we haven't got any previous, or we've got no drugs that slow down Parkinson's, but that's not for want of trying. This is just a small selection of trials which so far haven't shown any evidence of a neuroprotective effect. I've added the so far there just because Parkinson's UK have recently announced that they're going to be trying to take GDNF further forward following their trial, which people may have seen. There was a programme on BBC a couple of years ago about it, which was absolutely fantastic. A bit of programming to give an insight into people with Parkinson's and the GDNF trial. But so far, we've not found any kind of evidence of benefit from any of these drugs. So why is it so difficult to find a treatment? Well, essentially at the core of it, we still don't know why this happens. And that picture on the left is a pathological sample of the midbrain in someone with Parkinson's on the left and a healthy individual on the right. And that kind of black line you can see pointed to is the substantia nigra, literally the black substance. And this is one of the key areas that degenerates in Parkinson's and leads to a reduction in the amount of dopamine being spread around the brain, and that's what leads to a lot of the motor symptoms in Parkinson's. And we still don't really know why that happens. We know there's a lot of different aspects that tie into it. So mitochondria, which Simon's talked about in Alzheimer's, are also implicated in Parkinson's. Lysosomes are kind of recycling and breakdown machinery of the cells. There's evidence for inflammation in Parkinson's. There's environmental factors, and there's some genetic risk factors. But ultimately, in an individual, we still can't work out exactly why they have developed Parkinson's. And even if we do want to find a drug, well, how do we model Parkinson's to find a drug? And a lot of our research in the past has focused either on animal models of modeling Parkinson's and creating Parkinson's-like features in animals, or relied on genetic models of Parkinson's. Now, the kind of hereditary forms of Parkinson's, so if you inherit a faulty gene, you develop Parkinson's, probably only account for about 1 to 2% of all Parkinson's cases in the UK. But a lot of drug discovery and work has been done based on these genetic models, but they probably don't exactly mirror kind of typical sporadic Parkinson's and the mechanisms which may be going on there. So it may be that some of our models used for drug discovery have not really modelled Parkinson's exactly as we would expect them to. And then even if you do get a drug forward, how do you choose who to trial it in? If we take our whole population of people with Parkinson's, there's probably a broad range of kind of mechanisms which have gone wrong in their neurons to lead to the development of Parkinson's. And how do we select out those individuals? At the moment, other than genetic testing, we don't really have any tools to try and interrogate any of the other pathways or mechanisms which may be going wrong to direct potential drugs towards. And then even when you do start your trial, how do you measure Parkinson's? You can't get out a bit of string or a ruler. It just doesn't really work like that. So we've not really got any good blood biomarkers for Parkinson's, which either help kind of track the progression of disease or can easily be used to kind of easily quantify the state of someone's Parkinson's at a point in time. So we're kind of struggling on that side a bit. This is a picture of one type of scan. This is a DAT scan that labels the dopamine transporters in the brain. Now, DAT scans can somewhat be used to track Parkinson's, but if you run a trial based on that, there's an associated huge cost of performing DAT scans in people. It's a radioactive isotope that you inject into people to label these transporters, so you're exposing people to radiation. And actually, the number of people you have to enroll in a trial to detect a meaningful difference in DAT scans is really quite large. So there's a lot of challenges there on that side as well. And then can doctors measure Parkinson's? So we have lots of clinical scales and questionnaires, but in many ways they're quite crude tools to try and capture the many features of Parkinson's and truly characterize the state of someone's disease. And part of the reason is that every individual with Parkinson's is very different and very unique, especially clinically, and probably their underlying mechanism as to why they've developed Parkinson's is probably quite varied among everyone. And so the various clinical scales may be quite good for capturing, maybe one person's Parkinson's who has a lot of tremor, for example. But say for an individual without tremor, the same rating scales may not quite capture their Parkinson's in the same manner. And there's usually quite a divergence between what clinicians consider important in measuring Parkinson's and what patients consider important in measuring in Parkinson's and what affects them. So, in summary, previous trials have suffered from poor translation of drugs identified in either toxin-based or genetic models of Parkinson's. We have great difficulties in stratifying and subtyping Parkinson's for kind of what we call precision medicine approaches, and we have poor tools at the moment to quantify Parkinson's and measure clear treatment effects from the drugs. So how are we trying to solve this? So essentially my PhD focuses on developing kind of more sensitive and specific tools to achieve those aims. And they're kind of brought together in this clinical trial, which is the UP study. So this is, I'll go on to talk about this in a moment, but this is using two kind of novel techniques to try and measure how someone's Parkinson's is changing in response to a drug, and that's especially with the sensor-based gait analysis, where we can measure very clearly how people are walking and lots of detailed parameters about that. And then we're also going to be trying to measure directly if the drug is having an effect in the brain directly. And that's using 31-phosphorus magnetic resonance spectroscopy, which I'll talk about shortly. So the UP study is a phase 2 placebo-controlled, double-blind, randomized control trial. We're primarily looking at the safety and tolerability of ursodeoxycholic acid in Parkinson's. So to talk a bit more about that, so why have we chosen ursodeoxycholic acid? So this was a drug identified in a large drug screen by Dr. Mortiboys and Professor Bandman a few years ago. And ursodeoxycholic acid appeared to have a very clear mitochondrial rescue effect in both genetic and sporadic forms of Parkinson's. And this was primarily tested in skin cells to start off with. Other groups have also verified that it appears to be neuroprotective, so it appears to be protective in a toxin-induced mouse model of Parkinson's. And the other advantage of ursodeoxycholic acid is that it's been in clinical use for decades, 30 odd years at least, for treatment of liver disease and primary biliary cirrhosis. So there's good kind of safety and knowledge on the drug already, which makes it an ideal candidate for a drug repositioning strategy. So taking an existing drug that we have good knowledge and experience with in one condition, and moving it to another condition that we suspect it will have a beneficial effect in. So this is a very large table going into a bit of detail about some of the aspects of the study. So in total, we have 30 participants. We have 20 in Sheffield and 10 in London. People will be taking ursodeoxycholic acid for 48 weeks at 30 milligrams per kilogram or matched placebo. We randomise in favour of drug to placebo, so we actually will have 20 people taking the drug and 10 on placebo. We titrate it up slowly in the first month to try and avoid some of the side effects associated with it, and then it's followed by an 8-week washout period after the 48 weeks, where we then reassess people when they've not been taking the drug for a while. This trial is predominantly focused on people with early Parkinson's, so less than 3 years from diagnosis, and more mildly affected. And our primary endpoint, so the main outcome of the trial, the main kind of reason it's been designed in this way, is looking at safety and tolerability. So 30 milligrams per kilogram is about double the dose of what we normally use in clinical practice. And so we want to make sure that it is tolerated by people with Parkinson's. But things that we're very excited about are our secondary endpoints looking for evidence of a disease modifying effects of slowing it down. So that's looking at the sensor-based quantification of motor impairment, and the Movement Disorders Society Unified Parkinson's Disease Scale is our standard clinical rating scale, which I've slightly disparaged earlier on in the presentation. And we're also assessing for target engagement of UDCA. So that's to show that the drug is having a meaningful physiological effect using the phosphorus magnetic resonance spectroscopy. So this is the sensor-based gait analysis. So this is up on our clinical research facility. Some of you may recognize this is Professor Banman giving it a bit of a test run. He will rue the day he ever allowed me to film this, because I show it at every opportunity. But this shows what we have. So we have some strips along the side with lots of LEDs that can track the step width, step length, the amount of time people spend on one foot, and we have various sensors on the legs, on the head, on the back, and around the waist, to measure how people are rotating through space and moving. And essentially, this will give us a more sensitive tool to assess how people's gait are changing in response to the drug. Because essentially the clinical rating scale, the gait assessment, which is a huge part of Parkinson's, is essentially me watching you walk up and down a corridor and giving you a score out of 4, which isn't a particularly sensitive tool, to say the least. So in addition to the clinic-based assessment, which I've shown in the video, people also wear this kind of home activity monitoring device for 7 days. This also captures a lot of information about their gait and gives us an idea about how well people are moving in their kind of real life environment rather than in a clinic environment. So we're very interested to see if this can detect any differences in gait, and slightly more sensitive than what I can detect. And then to talk about our other aspects, so this is the phosphorus magnetic resonance spectroscopy. So this is a MRI technique experienced exactly like a normal MRI scanner. However, rather than taking just anatomical images, as you can see on the left, we also do something called spectroscopy, which is where we excite nuclei within the brain, and in this case it's phosphorus nuclei, and then when we excite them, we can measure how long it takes them to kind of relax back to their resting state in the scanner, and we can measure that kind of relaxation and decay back to resting state to infer the relative amplitudes and quantities of these compounds in the brain. So the ones that we're most interested in are adenosine triphosphate, so that's ATP, that's the primary energy carrying molecule in the body. That's super important. This is what your mitochondria produce bucket loads of. Phosphocreatine is an alternative high energy store, where you can break it down for short bursts of energy when needed. And then inorganic phosphate is free phosphate, which is usually released from the breakdown of ATP or phosphocreatine. And this is what a process spectra looks like, and we can analyze the amount in these areas of the brain. And the ones I've highlighted in yellow are the ones we're particularly interested in. And so I have two projects that I do this in. So one is the UP study, which is the trial, but I also have another project called Fox project, where we have 35 people with Parkinson's and 25 healthy controls. And the aim of that project is to see if measures of mitochondrial function derived from this MRI scan will correlate with the same or similar measures of mitochondrial function in the skin cells. So I take skin biopsies like Simon, turn them, derive fibroblasts from them, and we can measure how well their mitochondria are working in the skin as well. And the work in the FOX project will kind of help us establish the normal or expected kind of values in healthy individuals. It also gives us a chance to look at how Parkinson's affects phosphospectroscopy. Are there relationships with age, severity of Parkinson's, how long you've had Parkinson's. It also helps us give us a bit of information about relationship between kind of peripheral mitochondrial functions, such as in the skin and in the brain. And ultimately, we want to see if we can subgroup people with Parkinson's according to their mitochondrial function. Can we identify people who are probably going to be the most likely to benefit from mitochondrial rescue drugs? And then that gives us the information needed to then transfer our knowledge into the UP study, where we scan people before they take the drug and after they finished it, to see if the drug is having a meaningful effect. And ultimately, what are we wanting to move towards? Essentially, precision medicine in Parkinson's. So this is similar to kind of the picture I showed earlier. So we have a whole population of people with Parkinson's, and then using some of these techniques, things like the MRI scans, possibly some the cell readouts, blood biomarkers, and then also possibly some clinical assessments. And we can help stratify people into groups. So we can say, maybe, for example, your Parkinson's appears to be driven primarily by mitochondrial dysfunction, and therefore a drug such as ursodeoxycholic acid may be the best option to try and slow down your Parkinson's. Although hopefully we'll have options for lots of different types of Parkinson's. Maybe if you have lysosomal dysfunction, maybe it's this drug, and so on. So that's really in kind of the Parkinson's research community a big goal at the moment, moving towards precision medicine techniques. So in terms of the UP study, so we completed all our study visits in December 2020. We're currently in the monitoring phase of the drug trial at the moment, and hopefully shortly beginning the analysis phase of that. We're hoping to publish our results by June this year. We're very confident we're going to meet our primary outcome of safety and tolerability, and we really didn't run into any significant issues during the trial. It was very well tolerated on the whole, so we're very confident we'll meet that. And we're very excited to see what the sensor-based and MRI scanning parameters kind of come out in the analysis. And this is just to thank everyone. So Professor Bannerman's my primary supervisor, but I have obviously a huge amount of support from both Dr Mortiboys and Dr Tom Jenkins as well, who supervises me on the imaging side as well. There's a lot of people involved in these projects, and we have a lot of funding as well from a lot of charities. So JP Moulton Foundation and Cure Parkinson's Trust for the UP study, and then for the FOX project, Michael J. Fox Foundation as well. So huge thanks to everyone, and also to every individual who's been happy to take part in our projects as well. Thank you.
Thanks, Tom and Simon. That was really, really great presentations. So I'm going to coordinate the question and answer session, which we'll have for the remaining time period. And thank you to everybody who submitted the questions before the seminar, and I can see we've had some coming in throughout. So I'm going to kick off with a question to Simon, which was submitted by Cecilia before the seminar, and she asks, Simon, are type 2 diabetics more prone to develop Alzheimer's disease?
Yep. So I think the short answer is yes, but the longer answer is that the relationship is not that if you're going to get diabetes, if you've got diabetes, you're likely to get Alzheimer's disease. And understanding how sugar metabolism in diabetes is affecting the body as a whole is something we don't have a full grasp on yet. Potentially the brain itself determines the levels of people's blood sugar outside the process of diabetes. So the relationship is complex. Alzheimer's disease, diabetes affects multiple organ systems as well, and for instance, the integrity of the blood vessels that go to the brain, or how well they're maintained, which can be affected in diabetes, might be what's contributing to the Alzheimer's disease. So you are at an increased risk, but that doesn't mean that if you have diabetes, you're going to get Alzheimer's disease. And also, if you control your diabetes very well, that's probably the best protection that you can have against it.
Great, thanks, Simon. So I'll switch now and come to you, Tom, for one of the questions that was asked beforehand. Tom, Rafi has asked, is Parkinson's mostly a genetic issue? How many young people are diagnosed with Parkinson's?
So this probably varies slightly across the world, so I'll probably focus primarily on what we know in the UK. So Parkinson's can affect any age. The youngest patient we have in clinic is 29 at the moment, so it can affect any age. Probably only about 5% of Parkinson's occurs before the age of 50, and vast majority of it happening later, usually from 60s onwards, but it can affect kind of any age in adulthood. In terms of genetics, so probably about 10% of Parkinson's is related to genetics in some way. So probably about 1 to 2% of that is kind of familial inherited forms of Parkinson's, where you've inherited a faulty gene, and so therefore you have developed Parkinson's. So that's probably about 1 to 2%. The other 7 to 8% is probably related to genetic risk factors. So you have a certain form of a gene, which doesn't mean that you will develop Parkinson's, but does increase your risk of developing Parkinson's. So it's primarily not genetically related, but there are certainly some key genetic factors involved in it.
Thanks very much, Tom. I'll switch back with some more quickfire questions to Simon. So another one that was pre-submitted from Francis asks, is there any treatment to prevent deterioration when it's obvious a person is starting to suffer with dementia?
So treatment is very limited in all the forms of dementia, but we have, there's a group of drugs that are called the acetylcholinesterase inhibitors, and they work against is a particular group of neurons that seem to be affected in Alzheimer's disease. What they seem to do is slow the progression of the disease in a certain group of people. So anyone that presented with Alzheimer's disease to the clinic, we'd probably give them one of these drugs, unless there's things in their past medical history that would stop us giving them. So there is a couple of drugs which reduce the progression of the disease, but there's none as such that stop it at the moment.
Thanks, Simon. And I'll go back to Tom for the next question, also pre-submitted. Tom, David asks, what prospective drugs, in your opinion, have the best chance of providing an impact to slow down progression of Parkinson's?
So this is, I liked this question, but I don't have a good answer for it because, it's so hard to know when stuff is still in trial. I think there are hugely promising leaps being made in that over the last few years, we've had an explosion really in the amount of drugs going to Parkinson's trials and the amount of kind of drug development in this space. And so I think the outlook now compared to 10 years ago is probably a lot more positive than it was then. I think, there's lots of drugs at the moment, things like exenatide, which is a diabetes drug also being repurposed in Parkinson's. GDNF, which I mentioned in my talk, is now been taken forwards further by Parkinson's UK with the aim to maybe try and work out if that was having a beneficial effect a bit more. There are promising drugs focusing on kind of modulating the immune system and certain immune targets that may be beneficial in Parkinson's. Other drugs which are targeting alpha-synuclein, which is the main protein that aggregates in Parkinson's. And there's lots of stuff going on there, and I personally wouldn't want to bet on which one is most likely to be beneficial, aside from the fact that I have some obvious bias anyway. But I think it is hugely exciting at the moment, and yeah, I'd be optimistic of the state of research in Parkinson's at the moment and moving forwards.
Thanks very much, Tom. Yes, I completely agree with you. So we'll move on now to some questions that have been submitted during the live webinar. So the first one for you, Simon, is from Tilly, and she asks, can non-CNS glial cells from Alzheimer's patients be used for cell culture rather than using the astrocytes derived from fibroblasts?
So the answer is yes. We can use basically any type of cell really that we want to reprogram. Quite a lot of the reprogramming of making different types of brain cell is done through blood cells. So different types of white blood cell. So it's just we use fibroblasts because they're an easy cell to use, and there's an easy source and they're easy to access as well. Is the main we use, but yeah, we can use different types of cell to answer this question. It depends on what question you want to answer, really.
That's great, thank you very much, Simon. So I'll come on to Tom for an answer to this next question first, and then I'd like to flip it over to Simon to have an answer as well for the question. So this has come in from Hugh, and he asks, how easy is it to keep abreast of research being undertaken worldwide to ensure that there's not duplication, or is duplication sometimes good?
So I think from the perspective of kind of trial participants, and I'm going to answer this predominantly from Parkinson's perspective, there are some great ways to keep track of kind of either research that's being done or research that you can take part in. So Parkinson's UK, sorry, my cat's joined me now. She's keen to answer this question as well. So Parkinson's UK has an excellent portal on their site where you can look for research that you can get involved in, and you can put your postcode in and see what's going on around you. The Michael J. Fox Foundation also has excellent resources about trials going on. The Cure Parkinson's Trust, the Deputy Director of Research there, Simon Stott, has a fantastic blog on the science of Parkinson's if you're interested in kind of digging a bit deeper, and he keeps a very close eye on all the trials and developments going on in Parkinson's. So there are some fantastic resources out there. Duplication, I think, is very helpful. I think reproducibility of research is something that's very important, and I think also across the world it's very important to maybe look at these trials in different settings. You know, in the UK, predominantly our cohorts are Caucasian, and so a drug which may be beneficial in the hands of a UK cohort, which is predominantly Caucasian, may not necessarily be the case elsewhere in the world. So I think duplication in different populations is certainly not necessarily a bad thing.
Thanks, Tom. And if we can flip that over to Simon for his response.
Yeah, so very similarly, I think it is difficult, if I'm honest with you, because there's so much research that's done around the world now. You have to, so PubMed, which is a research website, there's ways that you can link things that you're interested in to get notifications when new articles are published. I think for participants in dementia research, there's the Dementia Research Platform website and the Alzheimer Research UK, Alzheimer's Society website. They talk about a lot of the big trials that happen. Twitter is sometimes a good, it has both pros and cons to social media, but there's certain websites with there's certain accounts on the Twitter website which are really useful to follow. It's one of the things I try and explore on my Twitter webpage as well. But yeah, I think there's ways of getting around it, but it is complex. And reproducibility is definitely, or repeating results is definitely important. So I think, yeah, we do need to do that. Yeah.
Thanks very much, guys. So we'll now close with our final question and we'll go back to Simon first of all for an answer on this one, and then if we have time, we'll flip it over to Tom also for a response. And this one has come in, and Eleanor asks if acetylcholine metabolism is important. Is there any connection with myasthenia gravis or another AC?
So not really in Alzheimer's disease. They affect very different parts of the nervous system. Myasthenia gravis is caused by a sort of an antibody-mediated reaction, whereas Alzheimer's disease, it's that the particular cells that have the acetylcholine start to degenerate, and that's what caused the memory problem. So there's probably no link with the two.
Okay, thanks very much, and Simon. I think we've finished bang on time, so I can hand perhaps to Andrew for a close.
Heather, Tom, and Simon, thank you very much indeed. I'm so sorry this is all that we have time for this evening. Now, I'd like to say a huge thank you to our speakers for the amazing work that you do, which we've heard a lot about this evening, and also for putting the time in to deliver such fantastic and accessible talks. Thank you also to the alumni team for pulling the event together, and for all of you for giving your time to attend this evening.